Hello, everybody. Again, my name is Ellen Dow, and I'm a biologist by training and part of the KBase user engagement team. I'll be leading this introduction to KBase as well as a tutorial. And with us is Elisha Wood-Charlson, who is the lead of the user engagement team here at KBase, and she'll be helping with some logistical support as well as speak to more on collaborations with KBase. So before I really launch in and get started, I want to acknowledge the land that we're on today. I want to acknowledge the traditional owners and custodians in all nations and honor their connection to the land, water, biodiversity, and culture, and pay our respects to their elders past, present, and future. The land that I work on is the ancestral and unceded territory of the Ohlone people in the Bay Area. Particularly, I work at Lawrence Berkeley National Lab. And I also live here in Berkeley. So what are we going to cover today? As an overview, I'm going to provide an introduction to what KBase is, its broad functionality, and then how KBase supports its researchers. So why would a researcher or why would somebody like you want to use KBase? And then I'll go into how you work into KBase with a tutorial. We'll walk through some demos about how the narrative itself works, and then we'll go into more on how KBase empowers collaboration between ourselves and you and your collaborators. So again, the question and answer document is in the chat. Please refer to that, pop in your questions. There are also some really helpful links, including links to the tutorial narrative that's not quite public yet, but it will be during the demo today. And there's also links for our YouTube channel, which has some really helpful webinars and how-tos. So you can subscribe to our YouTube channel and follow us on Twitter as well for any upcoming news and events that we have. Okay, so I have a few questions for you all before I start answering your questions. They are going to come up within the Zoom poll. So the first question is, have you heard of KBase? If yes, if you have, please let us know or you've heard of us in the chat. The second one is, how comfortable are you with command line tools? And then the last question is, what kind of bioinformatic analysis are you most interested in? If there's something that's not listed here, please, again, let us know within the chat. I might have to answer as well.
Go for it. So hopefully everybody can see and respond to the poll. I see answers coming in. If you can't respond to the poll, feel free to use the Zoom chat. And again, if you have heard of us, there's a couple of people that had heard of us, though that might just be Ellen and Roy, who both work at KBase. There we go, we've got a few more people. Please let us know how you heard of us. To JJI and Doug, excellent. Thanks, Doug. JJI, great.
Well,
yeah, I recognize a few friends. Go ahead, Em.
Yeah, we work a lot with JJI, so I see a lot of those folks on calls here.
Mm-hmm. All right, I'll give it another couple of seconds. Looks like most people have responded. And then quite a good breakdown on the kinds of analysis people are interested in. Okay, so I'll end the poll, and I think everybody should be able to see it now. Okay, so that's where we are. So it sounds like quite a few of us have not heard of KBase before, so welcome to this. Looks like many of you are very comfortable with command line tools, which is great. There's a few people that have some experience, but maybe not proficient. And then there is a pretty even spread about what kind of analysis people work on. So that's really great information.
Thank you. Thank you all for answering. Yeah, and as Roy said in chat, KBase and JGR are neighbors at Berkeley Lab up on the hill. So I'd like to talk about, first, especially for those of you who have not heard about KBase, what is KBase? So the Department of Energy Systems Biology Knowledge Base is a free open access software and data science platform that enables users to upload their own research data and analyze it alongside data from collaborators and within the context of public data. So the narrative platform itself is built on a Jupyter Notebook instance, and this serves as a bioinformatics lab notebook that gives researchers the ability to develop their data analysis pipelines, and then they can annotate and share these analyses. So functionally, how this system supports science and why you may want to use it. I know here a lot of the people are really comfortable with coding, but for those biologists who aren't so comfortable coding or are using the command line, using the wrapped tools is a really great feature. There's also the ability for data access. So whether it's data that is from your collaborators that you're sharing, or public data, so data that does come from the Joint Genome Institute that's public, or NCBI, if you want to access that and compare your data with it, you can do that all within the system. And there's also so many tools that are just in one place, so you don't have to go around or download everything onto your computer, and also accessing compute power in case you don't have that resource available at your hands. That is accessible through KBase. And so I want to turn it over to Elisha real quick to talk about where we are going with the platform in the future.
So this slide really is to kind of get to that future plan of like, why do we need KBase? Where are we actually headed with this? In part of integrating a lot of these different components of it, we are supporting what are called the FAIR data principles. So findable, accessible, interoperable, and reusable. In addition to those FAIR principles, we're also setting up reproducibility. And so Ellen will show you the KBase narrative, and it's built on top basically of a Jupyter Notebook instance. It also allows us to actually look at data and make sure that they are comparable. As many of you know, depending upon how the samples are collected, how they're processed in the lab, really kind of determines whether you can actually compare downstream data products and get reliable answers for them. And then understanding how to actually put these diverse biological data types in one system where they can actually be integrated in a way that's meaningful. And so tying all those threads together really starts to enable our vision for what we're calling the KBase Knowledge Engine, or KE. The idea behind that is that based on connections that are inherent in systems biology, KE and the infrastructure that KBase is built on can create a scenario that takes the contextualized data that's already in the system, along with evidence and reasoning behind it, and then present that to users that are using our system. So for example, someone comes up with a new genome they maybe find out in the environment, and the KE system would be able to eventually be able to say, okay, you found it in this environment, but it might also be present in environment A, B, or C. Maybe only when organism X is also present, because your genome is actually missing the full metabolic pathway to produce nutrient Z. And so being able to combine that systems level biology network across all of the data that has evidence already built into the system, we can start to do really interesting predictions. And so that's where we're headed with this concept of the KBase Knowledge Engine. Back to you, Ellen.
I am going to start to launch into the demo of using the narrative. So to start off with, here is kbase.us. This is our external facing site. So this is, if you Google KBase, this is likely what you're going to find. This is where a bunch of information about KBase is, as well as resources to use KBase. So we do have this Learn panel, which is about using the platform itself. We have a research tab that talks about collaboration and work that we're currently doing and that we have done. There's also the Engage tab, which, well, now it's showing as a previous event, but there's our event that we're here doing now, and any upcoming events. So if you're interested in learning more about KBase or coming to another webinar, you can check us out there, and news, so for the latest that has happened. To get into the platform, you need to have an account, which is free. You would go here up to the upper right-hand corner and click sign up. Or if you already have a KBase account, you can log in. Now the login is with a few different third parties. So we use Google. and ORCID ID, which is really great for linking publishing, as well as Globus. So if you are transferring a lot of big data, that's helpful to link to your account. And you can link them all within KBase. You don't have to just use one of these. You can use multiple of, you can link multiple accounts to your singular KBase account. So I can go here to log in, and I'm already logged in, so it's just going to go straight through. I'm not going to see the sign up sheet or sign in page. So it's going to pop open, and we're going to land on the Narratives Navigator. That's what first opens up when you launch KBase. And here is my narratives page. Again, if you're wanting to link different accounts, so your ORCID, Google, Globus account, you can click here onto this account tab and go through the process of linking those different accounts to your KBase account. Here is the Narratives Navigator, and I have my list of narratives. So all the narratives that I have created, going from most recent to older narratives. I can actually search with these as well as organize them a bit differently. And if I want to update something or something's changed, I can refresh it. This is especially helpful if someone shared a new narrative with me. As I said before, we have tutorial narratives, which are accessible from the Learn page. They're also accessible here within narratives. So these are public narratives that have been created by the KBase team and have been made public. There are also narratives that anyone has made public. So the default is for private for these workflows. And if someone has shared one publicly, you can find them there into that public tab. So I want to show you first what a new narrative looks like versus a narrative that I've already populated with the workflow. Just so you kind of see what it is before there's data and analyses within the narrative itself. So to go through, I open a new narrative, click on that new narrative button, and here I have a welcome to the narrative cell. So where this cell is sitting is what's built on more of that Jupyter Notebook instance. So this is where we're going to have data and app cells populate as well as any results. When I first come to the narrative, and if you're not familiar with it, you can click on our documentation tabs. So here, Quick Start Documentation goes to our documentation site, and this is docs.kbase.us. Here we have a quick start guide, which actually goes through how to sign up for a KBase account or how to sign into KBase, and goes through and talks about how to create a new narrative and how to use the narrative. In addition to this guide, there is a more thorough guide, which is the Narrative User Interface Guide, which is here. There's a lot of tabs, which each have separate pages on different tools. So there is how to sign up for an account here in our documentation site. What else is here within the narrative is we have over here a markdown cell. So this is a cell where we can add markdown or HTML language to spiff up our narratives, and I'll show you more on that. This is really where we get down to the annotation. There's also the ability to add a code cell within the narrative. And then over here on the left-hand side, here we have the data panel. So currently I have no data in this narrative. If I wanted to add data, I click on this blue add data button. And below that, we have our app panel. And this is essentially a catalog of applications that we have available. And these are all tools that have been wrapped as apps for the KBase UI. So for today, I've put together a narrative that shows in practice what a workflow looks like. So we're going to look at a metagenomics workflow, and this has some pretty neat data in it. So this, I will make this public, but this is the narrative that is linked within the Q&A document. So here, in this narrative, what we are working on is data that is is metagenomic data that is from the amberjack blue hole off the coast of Florida. So a blue hole is essentially an ocean sinkhole that's in the bottom of the ocean floor. And NOAA funded a group of scientists to go out in 2019 and scuba dive to 106 meters down and collect some water samples. From these samples, they gathered metagenomic data and sequenced this data using Illumina MiSeq sequencing with 2x300 base pairs paired end protocol. So this is important information for us to know when we're going through and processing these raw reads. There's a few different ways to get the data into a narrative. One of the ways is to import reads from the web. So here I have used an app which I directly imported these reads from where they're kept in Figshare, where they're stored in Figshare. But there's some other options as well. So you can also import public data from a public narrative. And here is actually where these the authors of who went out and gathered this data and wrote this, did the sequencing of this data, had gone through, and they actually put it in KBase. So this is accessible to anybody. And they went through and they added the data. And here you can see we have assembly set of all the metagenome assembled genomes, as well as a lot of assemblies for different depths and different data sets. So here at the bottom, here we have some of the paired end libraries that they had imported. So these are also possible to import into a narrative. And we do that by clicking on this blue plus button. So before, when there was no data, it had just that add data, and now we have a blue plus because we have data within this narrative. So here I click on the plus button. This is the data that I have. These are all my data, even the data that's not within the narrative, which I have a lot of data in this one, so it takes a little bit to scroll. But here's some data that's not in this narrative. I also have access to data that has been shared with me that I can add to the narrative or access to public data. So these are all the public data sets. So I could go through and I could search for the public data that was within that narrative. These are all organized. So we have NCBI, RefSeq genomes, public media ontologies that are available here. There's also example data if you're ever just interested to go run a play just to see what a data set looks like in the narrative. And we have everything from reads, assemblies, genomes, flux balance analysis models, media. And then there's the import tab. So this is if you're importing data from an external source, so outside of the narrative. So you can I had mentioned before that you can use Globus for transferring. You can also upload with the URL. So if I were to click the upload with URL, it would create that new app cell that I showed you of how I imported these reads using the URL link. The other option is to select from my computer or drag and drop. And I figured I would show you a little drag and drop. So I have some files off screen, and I am just going to select one of those files and drag it over and drop it. And so now this file is currently uploading to my staging area. And this staging area is sort of a holding place for data. So this data will only stay here for a limited amount of time. It'll be deleted after 90 days. But for now, it can stay there. So when I'm ready to import this data to this narrative, let's see. I make sure that the data type is selected. So yes, this is a GenBank genome. And then I would click here and have this selected, and I could import the selected. One of the new functionalities on KBase that we just released is bulk import. And this way you can import multiple files at once into a narrative. So all of these files that have been uploaded, I can go through, and it's really nice. They have the suggested types. So I can click through which type of data file it is. This one's goes with the GFF metagenome. And I can go through to different pages and select the files that I want to import, and then I would import selected. And this would spin up a new data import cell. However, I just want to go over that this is currently only functional for, let's see, in my handy docs guide, it's only functional for certain data types. So if I scroll down here, you can currently use this with assembly FASTA files, SRA reads, FASTQ reads, both non-interleaved and interleaved, GenBank genomes, and GFF metagenomes, so the FASTA and GFF3 files. If you were to import or select a file type outside of these, it would spin up a few import cells. So you wouldn't just have that one. And because I have, because I have these selected, I can just hit import selected so you can see what the file with the app cell looks like. So this one, I was only opened up one because these were all the selected types. My assembly file already has the information it needs filled in, but I can edit the parameters. Here I have a little warning that I need to fill in parameters. So I can do that. Oops, if I can spell single. So I can do that. And when I do, it gives me a little green check mark that is good to go. And once I filled everything out correctly, which I'll just show you. Right now I can't hit run because I have not done that yet. Yeah, GFF. So I could hit run and this would start an import here. So those are a few different ways on importing data. Again, all the information on how to import, all the different ways, methods you can import data are described within our documentation. And so now that I have data in my narrative, because I already preloaded this like a cooking show, we can go through and analyze these raw reads. So the main question that we have is, how are we going to analyze this metagenomic data? So the metagenomic data is the information that's about the whole community of organisms that are there. It's sort of a snapshot of what was collected at that moment in time at the amberjack blue hole at 106 meters deep in 2019. So these raw reads, we first need to process them, and then we can assemble the genomes that are there. I want to figure out what organisms are present, so I can do some read-based taxonomy. And some additional research or some additional analysis that I can do is binning. So I can take those reads, group them. So theoretically group the different genomes into what could be considered a collective genome or that metagenome, assembled genome, and also assign taxonomy based on those genomes. And that sort of helps them with the completeness of each of the genomes that are there. So first with read processing, I'm going to use True-O-Matic. So if I look at my data panel over here, this is the original paired end library. And I clicked on this little 3-dot menu to expand the information about this data object here for the paired end library. So if I want to analyze this data object, what I can do is I can organize the applications I have and see what applications will take this data object. So if I click here on show apps with this data object as the input, this will organize my data catalog down here. So first I'm interested in read processing. That's the first step I want to do. So I click on that arrow to look at the different apps that are available for read processing. And these are a bunch of different apps that are available. So I'm interested in trimming the reads first. So I selected Trim-O-Matic, and that populated a Trim-O-Matic app cell within my narrative. So what I did is I needed to select the input object. So I selected that read object, and then I set my parameters. I knew that this was aluminum iSeq, so I selected the Nextera adapters to trim. And then I left pretty much everything just as it was for the standard parameters, the default parameters. And then I gave my object a name. And so for the result with this run, I had a few objects that were created. I had my paired in library, and then I also had forward and reverse libraries that appeared. And along with the objects that were created that I can now see within my data panel, I also have some results. So some information about the input, what was surviving, the surviving reads, the surviving data, any data that was dropped. So now that I have my trimmed reads and I've clipped off those adapters, what I want to go through and do is assess the FastQC, assess the quality using FastQC. So I did sort of a similar step where I went through the apps and I wanted to do some assessment of quality. So I could also search here, FastQC. Again, it's under read processing, select that app. Instead of choosing the original data file, I selected the trimmed data file. And with this result, when I ran it, it does not create a data object. Instead, I solely get a report. And with this report, I can also open it in a different window so I can see it a bit more clearly, zoom in on any of the information. I see here, basic statistics are good. I have a bunch of sequences. Yeah, sequence length is 36 to 301. I had 300 base pair read length. GC content is at 48. Quality looks good overall. And I also have some different information that the FastQC report gives me that I can check out. So now that I'm satisfied with the quality of my reads after trimming them, I can go through and start assembling these reads into contiguous regions of DNA or contigs. So I want to kind of examine what each of the genomes are here, or put them together so then I could go forth and figure out what different organisms I have. So there's a few different tools to assemble reads into contigs that you can use in KBase. I did 2, MegaHit and Metaspades. And beyond using this app catalog over here to search, so I could just go here, look at assembly for genome. Because this is metagenomic data, I could also click on microbial communities and then look at the microbial communities here. But beyond looking within the app catalog, I can go to the app catalog that is accessible from the narrative. So this will look familiar. This is where my narratives were. I clicked on the app catalog, and then I was able to open the app catalog here. So I can organize them in a bunch of different categories, types. Right now, it's pretty much categories. So I can go down to genome assembly to look at the different assemblers here. I can also scroll down and look at what the community apps are and what assemblers are available. So I have IDBA is one of the assemblers, MegaHit, Metaspades. I think that is it. That is it for the assemblers that we have for microbial communities. And the other app catalog that we have is if you're out of KBase, so you're thinking about, okay, do I want to go into KBase and start a narrative, is we have an external app catalog, which is kbase.us/app list. And here, these are all of the released apps that we have. So I can go down again to microbial communities and look at the released apps. And here I can see, yep, IDBA is there, Metaspades, MegaHit would also be in here too. Yep, right there. So similar to what I had done before, I selected my input object as my paired end read library, set my parameters. I could have done large or sensitive. I decided to do the quick and fast because I had a very large assembly of like a large set of data. And then I chose the output name, ran this application, and as the output, I received this quality report. So here I have the number of contigs that were assembled. I have the information about what the largest contig is, some of the some more statistics about what was present in this assembly, as well as a summary. Metaspades, very similar. Chose the trimmed paired end library as my input, and then gave my output a name, just went with our standard parameter result. I get this quality assessment tool as my report along with the object. So here I have a Metaspades contigs, and then I have the MegaHit assembly that was created with these two runs, and I have similar statistics for my output. So here I can actually see that there's a few more contigs that were created from the Metaspades versus the MegaHit. So with MegaHit, I had less than 8,000, and with Metaspades, I had more than 8,000. And then I can also see these assemblies within my data objects. So now that I have these assembles, I've assembled these reads, I would like to go through and assign some taxonomy. So figure out what different organisms are present. And for this, I chose a few different apps. I chose Kaiju and Gotcha. I also wanted to show how we get to apps that are in beta, so apps that aren't fully released yet. So with Kaiju, again, very similar. You have to choose your input objects, select the parameters to get the result. And I really like the Kaiju output because I get some really pretty bar plots. And again, I can look at these outside of the narrative, so I can examine sort of what organisms are present from at the phylum level all the way down to if I can identify any of the organisms at the species level. Similarly, Gotcha 2 will also give me some taxonomic information back in a little different format. So here I have a list of a table, but I can also examine a dendrogram. So if I want to open this in a separate tab, I can zoom in and look at a dendrogram of what species are present. I can also look at the chronoplot of what was identified within my reads. So from those assemblies, I have this great information. And then there's also the option to use apps that are in beta. So I already ran Centrifuge, which is an app in beta. It's a really similar output to Gotcha 2. And the other one I wanted to show today was Kraken. So if I go over here to look at my apps that are already released and I search Kraken, that's not going to come up. What I need to do is I need to toggle over to our beta apps. And this is going to give me a big warning that I'm going to go into apps that have not yet been released. So these are ones that we're still testing. They are in production, but they're not quite ready to be released yet. And so here, let's see, I want to make sure, okay, I've selected this app. So when I populate this, this narrative with Kraken, it will go below that app. So I hit Kraken. There it was under microbial communities. I select the app name, and now I have Kraken in my narrative. And so here I would go through and I would select the paired end library or read set that I want to analyze. I hit Kraken 2, and then to run, I could hit run. And then that would kick off my jobs. And so if I was running a job, or if I ran into any errors and was trying to investigate what was happening with my job, I can go to, I'll use this one. I can go to the jobs tab, which is again here, if you go, so also if you're just in your narratives, you want to go back, I can go here to my jobs tab, and here you can see I've been working on this, this webinar for the most part. So I can go click on the little information button, and this will give me my job log. So this is all the information about what I've been running in my job log. If I have any errors that come up, I can actually find them here. And if I'm talking with help desk to get any help about issues that I'm running into, I can just copy the problems that I'm running into, so that I can say, hey guys, this is the error I'm getting when I'm running this job. What's going on? Give them as much information as I can so that they can help me figure out what the issue is. And so I wanted just to show a couple more steps in assigning these. So I have an idea of what the taxonomy is, but if I want to go to a slightly different resolution, I can also bin the contigs, so group the contigs together based on their similar sequence that suggests that they are genomes or that they're related to one another. And the example that I chose today was MetaBat2. That's what I decided to use for contig binning. For the result, I was able to create 14 bins here. And I have some output information. From there, I took those bin contigs, and I did a genome quality assessment with CheckM, similar to what I had done before with FastQC, and I was able to run an assessment for quality on my bin. So you can see here up top, I don't quite have a lot of information about, like in bin 10 and bin 1, but as I go down, bin 3 looks really great. Bin 5 is, I have information, but there is some, because I do have some missing information, but there is some contamination that's present. I can see based on this quality check. But there is more information versus what's missing. So what I can do is I can also run a filter now that I've assessed the quality and say, okay, like I want to rule out anything that isn't more than 80% complete or is contaminated, and run that and get some results back as well in a similar form. And once I've grouped all these contigs together into bins, so my prospective genomes, what I can do is I can then assign taxonomy. So similar to how we had to assign taxonomy to reads, I can go through and assign taxonomy to the bins. So with this, I decided to use GTD-TK, and I used as the input the metalarge bin as my output object. So these objects are always populating within my data panel here. If I ever want to check out what these data objects look like, I can drag them into the narrative, or I can double click too many times to open more tabs than I want to. And then if I need to, I can just delete a cell. And it says, do you want to delete that? Yes, I did. So here I have a summary of what's going on. So I can see, yes, I have 14 bins still, even with filtering. And here I have information. So like how many contigs are there and such. And if I want to just focus on a few different contigs, I can use extract bins as assemblies. And here I have a list of all the contigs. And I selected 4 that I wanted to pull out. So I pulled out bin 2, bin 3, bin 9, and bin 10. I don't know if you remember, but bin 10 was one of the ones that was farther up and wasn't quite complete. But bin 3 was more of a complete one. So I wanted to get sort of an idea. So I extracted all of these as one assembly object. And here I can see that up top. So I have all these assemblies, each separate. So if I wanted to go in and investigate each of these assemblies and figure out more on what these genomes represent, or sort of the metagenome assembled genome represents, I could investigate each of these on their own. But I want to just investigate the assembly set. And so I chose to use DRAM to go through and analyze a bit of what was here within these assemblies. And so with DRAM, I have some information about electron transport chain complexes. I can grab some information on nitrogen metabolism, what's really like what functionalities are present within these genomes. I can see that there is no photosynthesis happening, which is not surprising to me at 106 meters at depth. You're not going to have much light reaching down there. And how I know this is there is a little legend over here that says green, it's present, light blue, it's not present. So that's how I knew that we weren't able to find any genes representing any of these, any of the functionality we need for photosynthesis. But we can see that there is some functional categories or functional IDs for methanogenesis or sulfur metabolism and nitrogen metabolism. So it just gives me more information about functionally what's going on within the sample. And really, so that's one method of kind of going through and examining some metagenomic data. And now I wanted to talk about some of the new functionality that's in KBase, which is what we call samples. So samples themselves are, it's a newer functionality, so it's a new suite of features that link environmental metadata with experimental data. And really the aim behind this is to evaluate and pull microbial data together based on categorical metadata and then analyze it. And so here we're using metadata that uses standards that have been defined by the communities. So these communities include ESS Dive. We also have IGSN, CESAR, and Enigma. And so these have been defined as the information that is necessary to make data fair. So again, that gets to that findable, accessible, interoperable, and reusable. And the aim behind following those fair data principles allows data to be reused across different experiments and across platforms. And within our metadata, some examples would be the geographic location, temperature, collection information, environment, material depth. And currently the functionality is mostly associated with metabolomics or chemical abundance data matrices, as well as amplicon data or operational taxonomic units. So you can have amplicon data matrices that you analyze. And we have a bunch of information on this functionality within our documentation. So we have, we discussed the ability to import these sample sets. So there is information on how to format your metadata based on these different community standards. And then there's also some functionality. So for functionality, these are available under amplicon and data matrices for the most part with amplicon data. So if I wanted to go more into examining metagenomics data, I can look at metagenomics from that amplicon and OTU perspective. And so what is present there are is functionality based on filtering abundance, also using sample set metadata to help define what analyses you want to run, so define those categorical data that you want to compare. And the really great thing about having these current apps and functionality on systems is that you can track the processing of the data. So from the raw data through what processes it has gone through, and then through the analysis pipeline. And so before we, I also wanted just to show a few different apps that are here, and I did populate this narrative with some links, so you can see sort of importing the sample set of how to import the metadata with these apps, and then importing your amplicon matrix here. And this is pretty much everything I have wrapped up in this narrative. So if I wanted to share this narrative, first I want to make sure I've saved it, so it's the latest version, and then I can go to share, manage sharing. I can share with particular users, so I can search for my colleague Zach and give him different permissions, either view, edit, save, share. I'll tell him he can only look at my narrative, so I can share it with him. I can also share it with particular groups, so I can share it with different organizations. And I'll talk to you a little bit more about how organizations work. Or I can make it public, so anyone can access and view my narrative. And if I do that, I click here, and now it's public. And now you can access this narrative from the Q&A sheet. And then I also wanted to share from the narratives orgs. So if I wanted to share it with a particular group of collaborators that I'm working with, I would go to orgs, click on there. And then these are groups of collaborators or people who want to access a bunch of narratives together. So here's actually the KBase Educators org, and there are a bunch of narratives in here for education purposes. We also have the Microbial Dynamics School, which I belong to, and I can click on that, and this will open up the organization. And here we can see a list of narratives as well as members here. And so if I wanted to create my own narrative or create my own organization and share it with a particular group of people, I could click here on create an org. And this is where I can populate my information for my org. And I can invite certain people to my organization and share my narrative easily with a bunch of people at once, without having to make it public or going through and selecting each person individually. And if I'm ready to share my narrative as well for a publication, let's see, I can make a static narrative, which, because I've made it public, I can do that. I can go here and create a static narrative. And Elisha will go over more on that with publication. The last bit I wanted to talk about was a bit more about our community developed tools. So I had demonstrated the output of this tool, DRAM. And so DRAM was actually created in collaboration with the University Research Lab. It was Kelly Wrighton's lab. We also have other community developed tools that are using our software development kit and bringing new functionality to the system with other collaborations, such as one with a group of researchers that were involved in a Department of Energy science focus area. And this group created VirSorter, which brought some new viral functionality onto the system, which is something that they were really interested in. And so this sort of wraps up my part of the tutorial. And we have one more question, and then I'll let Elisha take it from there.
Awesome, thanks, Ellen.
Yeah.
So we're going to talk a little bit about publishing, but before we did that, we wanted to see how people are participating in sharing knowledge that you generate as you go through your analyses. If you have other options than what's in the Zoom poll, please put it in the chat. as we're talking about this, I do want to emphasize that KBase is based on open software, and all of our software is actually in GitHub. And so everything is open license. And we do support through Berkeley Lab, all of our personal publications are required to be open access, and we do try to support all of our users to publish in open access as much as possible as well. So what we're talking about with publishing with KBase, this is what Ellen had alluded to with the static narrative. So the static narrative, when you go through and you click static narrative, it basically takes an HTML snapshot of your narrative at that particular time, and it puts it into a searchable version that lives outside of the KBase login. So Google starts to index them. You can put that link into publications for review, for grant proposals, etc. You can contact us and we can actually get a DOI for those static narratives. So once you get it all nice and pretty and you want to put it in a proposal or a publication or a presentation, we can actually give you a DOI for that particular static narrative that you can use to share. The nice thing about this is it sort of solidifies the knowledge that you have in that reproducible workflow for sharing, and it doesn't require any of the reviewers to actually create a KBase login to go and look at it. Not all of the data are directly available, but they can click on it, make a free account, and then go through and actually look at your narrative directly there, copy that narrative, and start doing their own analyses. And so part of the goal of this is to take a reproducible workflow that somebody sees in a publication, and within 5 minutes, they've taken your narrative, made a copy, added their own data, and reproduced your entire analysis within a couple of minutes. So that's sort of the goal of what we're trying to do here. So you don't have to go to an online repository, download a zip file, open the zip file, try to unpack what all the data actually are, upload that into your server, and then start to run on command line, right? The goal is to actually streamline that and make it less than 5 minutes before you can actually start using the workflow in your own data. That version has been fixed in that HTML narrative, and we track that version, so you can continue working in your narrative if you would like to, and create subsequent static narratives, and we continue to version everything. There was a question in the chat or in the Q&A doc about provenance, and KBase tracks provenance for every object that created in system. So you can actually go back through and actually reproduce everything as necessary. So as an example, we're working with the American Society for Microbiology that has a data publication called Microbiology Resource Announcements. These tend to be genome announcements, metagenome announcements, very short publications that just announce data that people want to get out into the community, but get some sort of traction on. And we're actually creating a template for people to actually produce these reproducible narratives for their submission. And MRA is actually really interested in this, the editors at MRA, is because we can actually track usage metrics of those data sets in our system, because they don't currently get many citation metrics for just the data set. So that's a collaboration that we're starting to build out with ASM right now. Next slide, please. And so part of the reason we're really trying to create this linkages about data sets is we want to be able to measure impact of data, not just publications. And so this DOI that we can give, the static narrative, gets registered with DataCite. We can also map your ORCID to it. So ORCID is kind of the individual's persistent identifier, just like the DOI is the dataset persistent identifier. We put those two pieces of information and we can actually start to report back to DataCite, which is kind of the aggregator of all dataset DOIs. Crossref takes publication DOIs, DataCite takes data DOIs. And we can actually start to report to DataCite metrics of impact for your data in KBase. And so we're actually going through and saying, how many times have people viewed your data? How many times have people copied your data? How many times have people downloaded your data? And start to actually provide a separate kind of metrics for how people are working with the data prior to or in addition to a publication that you might have in the field. So that's the kind of infrastructure that's just starting to get put into place between Crossref and DataCite and ORCID and the DOI repositories as well. So if anyone is interested in that, this is still kind of a developing field. We're taking user stories, and I would love to talk to you to understand kind of what your use case might be in this area. Next slide. So that kind of wraps everything up on our end. There is a bunch of links in the Q&A doc. There's obviously the various different components that Ellen showed you, our main website, any of the documentation pages. this recording will go up on our YouTube channel for DOE KBase. Please follow us on Twitter. We put out announcements for any new features that are going out, any new training sessions that might be happening. Ellen runs our Twitter account, so she's pretty good at being active about that. Everyone that registered, we will take your emails and we will put it into our newsletter list so that you can get updates that way. We send out like 1 or 2 a month, so feel free to opt out if it's too much, but it's not something that we really spam people on. It's mostly just informational newsletters that go out that way. And then finally, Ellen is kind of our West Coast representative. So KBase is spread across 4 national labs. Berkeley Lab is, of course, the local one to this group, and Ellen is our West Coast representative. So Seattle folks all the way down to Southern California. Ellen is kind of our person for training and outreach. So if you need any other training information, if you have any, whenever in-person workshops start to happen, she's the person to sort of reach out to for that. And I'm kind of the go-to person for any possible research collaborations. We mentioned the software development kit. If anybody has tools that they would like to put into KBase, let me know. We can talk to you about getting training on the SDK. If you do see any possibilities on how we might collaborate and with some of the research that you're currently doing, definitely please reach out about that too. We'd be more than happy to continue to extend our network there.
Well, I just want to say thank you, Bay Area Bioinformatics Forum, for having us. This has been great. It's really given me a good opportunity to create this a demo to show you about the whole metagenomics workflow and show off some of the new functionality, such as the bulk import, and discuss just a little bit about samples.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.